Hello, my name is Jaden Martinez, and today I'll be sharing my Module 4 presentation where I'll be discussing cell division and inheritance.
Okay, first, here's a little overview or table of contents, going over all the topics I'll be covering in this presentation. So first, we'll talk about the cell cycle, and then we'll talk about an all-important checkpoint within that cell cycle called the G1 checkpoint. Then we'll go over the process of mitosis, as well as cover sex-linked inheritance, and then we'll be talking about chromosomal inheritance with a specific look at X chromosome inactivation. And then we'll finish off with a conclusion and then a few questions. Okay, first, let's go over the cell cycle. The cell cycle is a series of phases that leads to cell division. And in the middle here, we have a picture symbolizing the cell cycle. Beginning with the G1 phase, that is in purple. The G1 phase is characterized by normal cellular activity where no changes are specifically evident, and the cell is biochemically active. In the corner of the G1 phase, you can notice there's something called the G0 phase, where the cell is in a non-dividing and non-replicating state, basically at rest. The G0 phase is an alternative to the G1 phase, in which the cell has postponed dividing, postponed moving on to the S phase for the time being. And it could also choose to stay in this G0 phase for the rest of its cellular life and not grow anymore. Okay, the S phase is the next phase, and this is where the cell becomes committed to dividing. Um, it's also called the synthesis phase, and this phase is indicated by the green color. This is where the cell will enter this phase um, because it wants to divide and it'll replicate its chromosomes to form a pair of sister chromatids. These chromatids are then joined together at the centromere, the end result being that the cell will now contain twice as many chromatids as the number of chromosomes that it had in the G1 phase. Next, after the S phase, the cell moves into the G2 phase, which is pink as it prepares for mitosis. Uh, yeah, so here the cell synthesizes the proteins for chromosome sorting and chromosome division as organelles reproduce and the cytoskeleton breaks down and the chromosomes begin to condense. The G1, S, and G2 phases are known as interphase, or time of normal growth in preparation for cell division. Next, we move to the M phase, or mitosis. This is where the replicated DNA and cytoplasm of the cell are split, and the cell divides into two nuclei. This involves several stages that allows for cell division, as the pairs of chromatids are separated and sorted so that each daughter cell receives the same amount of 46 chromosomes in human cells. Mitosis is then followed by cytokinesis, in which the cytoplasm is physically divided to produce two distinct daughter cells. Now, we're talking about the gap1 checkpoint, or the G1 checkpoint. In this presentation, we'll be focusing on the G1 phase and the G1 checkpoint, which is also called the restriction point. During the cell cycle and during interphase, there are three internal checkpoints that help ensure that only healthy cells are replicated. One of these checkpoints is found before the transition from the G1 phase to the S phase, as shown in the picture on the upper right and where the blue arrow is pointing. That's where the G1 checkpoint is. At this checkpoint, the cell uses internal and external factors to determine if the cell is able to move into the synthesis phase. Basically, the cell uses this checkpoint to determine if conditions are favorable for cell division. Checkpoint proteins determine the state of these internal and external conditions. Some of these internal conditions the cell considers are the cell's nutritional state and its cell size. A few external factors the cell checks for are the presence of nearby dead cells, the release of growth hormones, as well as the amount of crowding by other cells. The cell will also check for DNA damage before replication, which is very key so that no mutated DNA or other problems are replicated in future generations. If everything looks good, the cell will enter the S phase and commit to dividing. The cell is regulated by a few different mechanisms that help facilitate cellular growth and progression into the S phase. Or these mechanisms help prevent the cell from proceeding and protects against damage and protects against mutated cells from replicating. First, one of these mechanisms uh, is seen through cyclin proteins that can help advance a cell through the phases of the cell cycle by turning on certain proteins and processes which are important for particular stages in the cell cycle. One of these is cyclin D protein, as it helps initiate DNA synthesis and replication. It does this by binding to cyclin-dependent kinases, or CDK, or CDK, and form cyclin CDK complexes. These complexes will then inactivate RB, or what's called a retinoblastoma protein, a negative inhibitor. By phosphorylating this RB, the RB releases a transcription factor bound to the RB, um, and the transcription factor allows proteins to turn on and genes to be made, allowing for further replication, DNA replication, allowing the cell cycle to advance. This process is depicted on, oops, the picture on the right, bottom right. You can see RB being phosphorylated and then releasing that transcription factor onto the DNA. Also involved is P53, which is another protein which prevents cells with DNA damage from proceeding the cell cycle. It does this by stimulating uh, protein P21, which inhibits cyclin and the CDK complex from helping the cell dividing. P53 and RB are known as tumor suppressor genes, as they can help stop the formation of tumors, since their function is to stop damaged DNA from replicating. Okay, so now if the cell has identified any DNA damage or any other environmental factors that doesn't like, it will prevent the formation of these cyclin and CDK complexes and stop the advancement of the cell cycle, meaning the cell will postpone S phase, sorry, and may re-enter the G1 phase or return to G0 phase and potentially never divide again. The cell can also address some of these problems that prevented its progression and fix any damaged chromosome or cellular feature like an abnormal organelle in order to later pass through the checkpoint and enter the S phase. Okay, next we're going to be discussing a phase of cell division, this being mitosis, as shown in the picture in the middle of the screen. Mitosis is a cellular process where cells divide in order to reproduce and replicate. One cell divides to produce two new cells called daughter cells. These daughter cells are typically genetically identical to the original cell. There are essentially five phases of mitosis and ends with the final stage called cytokinesis. The key importance of mitosis is reproduction, as this process allows living organisms to increase in numbers and replicate their genetic material. This process further allows organisms to become multicellular organisms and grow their various tissues, organs, and organ systems. This process is found in plants, animals, and fungi. There are five phases of mitosis, with it ending in cytokinesis. The first phase or stage is called prophase, where the chromosomes condense and show that distinct X-like structure you see in all the pictures. The nuclear envelope begins to break down, and organelles move to the edges of the cell, while spindle microtubules form from centrioles, and the nucleolus disappears. Next, in prometaphase, the nuclear envelope disappears completely. Protein connected core starts to develop in the centromere region, which attaches to the chromatids and the spindle microtubules. Oh, attaches to the chromatids and to the spindle microtubules. The spindle fibers from the centrioles then connect to the connected cords. Okay, step three, the cell enters the metaphase stage of mitosis, where the spindles line the chromosomes up along the center of the cell, along the metaphase plate, which is halfway between the poles of the spindle apparatus. The sister chromatids remain attached by cohesion proteins in metaphase. This phase prepares the chromatids to be equally distributed into two daughter cells. So continuing with the stages of mitosis, then from metaphase, the cell enters anaphase, where the connections between the two pairs of sister chromatids are degenerated, allowing for the chromatids to separate, like this. Each individual chromatid moves away from each other towards opposite ends of the poles of the cell. This process elongates the cell as well. And then the next stage is called telophase. During this phase, the chromosomes reach opposite poles and begin to decondense. The spindles also depolymerize into tubulin monomers. The nuclear envelope reforms and the nucleolus reappears. This portion of the cell cycle, or the M phase, ends with cytokinesis, where the cytoplasm physically separates, forming two identical daughter cells, thus ending cellular division, which you can see at the end of the picture. Okay, before moving on, it's important to know that there are differences between plant and animal cell mitosis. As you might recall, plants contain a cell wall, which animals do not have. The cell wall is so rigid that the plant can't manipulate it or reshape it to form new cells. Instead, the plant cells build a new cell wall called a cell plate during mitosis that will eventually join together with the cell wall to split the cell in half. The cell plate is built in the middle of the cell during mitosis, and you can see this happening in the bottom picture, the blue one, where plant cells are undergoing different stages of mitosis. But you can see in a few of the cells where the cell plate is forming during the mitotic phase. Okay. Animal cells also have different processes for splitting the cells during cytokinesis, different from plant cells. Animal cells use microfilaments to split the cell into two daughter cells. This actin microfilament forms a ring around the cell um, that slides over itself to constrict and create what's called a cleavage furrow as the cell is split into two daughter cells. Okay, now we're going to discuss binary fission, which is a process prokaryotic cells undergo when dividing and replicating. The main purpose being that one mother cell will split into two identical daughter cells. This process is essentially carried out in 5 steps. First being that the DNA is replicated at the origin of replication, which is the first part of the DNA to be copied. The two origins then move towards opposite ends of the cells, pulling the rest of the chromosome along with it, elongating the cell. Um, Next, in step 3, FTSZ proteins form a Z ring in the center of the cell. This ring then forms a septum, and the septum pinches the cell and forms two new cells. This is depicted in this image here on the right. So how is binary fission different than mitosis? Well, first, they're kind of similar processes in that they're both part of cellular division and cellular replication. Both processes, chromosomes are copied and separated to form new cells. However, prokaryotic cells organize their genetic material differently than eukaryotic cells. Their chromosomes are usually organized into a double-stranded circular DNA molecule that is found in the nucleoid region of their cell, while eukaryotic DNA is found in the nucleus. Additionally, prokaryotes have um, DNA structures called plasmids, which are depicted as little green dots in the image, which is just extra chromosomal DNA that exists separately from the main chromosome and can replicate independently of the main chromosome and offer some growth advantages to the main cell. But they are distributed to the daughter cells. Also, another main difference is that the DNA replication and DNA separation happens at the same time in prokaryotic cells during binary fission, whereas in mitosis, DNA is copied during the S phase, and then the DNA copies are later separated during the mitosis phase. So they happen at different times, whereas in prokaryotic cells, this happens at the same time, allowing for rapid division and rapid growth. Now we're going to talk about sex-linked inheritance, or how genes located on sex chromosomes are transmitted from parents to their offspring. Sex-linked inheritance follows the principles of Mendelian inheritance patterns and the chromosome theory of inheritance, in which chromosomes contain DNA, and this DNA is the genetic material that is passed down from parent to offspring. Also, sex-linked inheritance follows the principle that some genetic traits are dominant and some are recessive traits. Sex-linked genes are those that are found on one sex chromosome but not in the other, thus meaning that sex chromosomes can determine sex differences. Mammals follow the XY system of sex-linked genetic inheritance, in which females contain two X chromosomes and males contain one X chromosome and one Y chromosome. Many recessive X-linked alleles cause diseases in humans and are more common in males than females. This is because male offspring inherit only one X chromosome, and thus they may receive a recessive disease-causing allele from their female parent. As stated earlier, the process of sex inheritance follows similar Mendelian patterns of inheritance and can be understood and predicted using a Punnett square like the one on the right. So this Punnett square that I made is an example of sex-linked inheritance. Um, in bears, where fur color is a sex-linked trait, um, the dominant trait with a capital B is blue colored fur, and the recessive trait is purple colored fur with a lowercase b. So this Punnett square um, shows the combination of a blue male and a blue female and their estimated offspring. So as you can see, most of the offspring will have blue fur, while one male will have purple fur. This is because this male will receive a recessive gene from the mother, which is found on the X chromosome. And expanding further upon sex-linked genetic inheritance, it's important to learn more about the variations in this process that may impact genotypes and phenotypes. An example of this is an epigenetic change called X chromosome inactivation. X chromosome inactivation occurs during embryonic development in female mammals. One of the two X chromosomes becomes inactivated during development and then forms what's called a bar body, which is when this inactive X chromosome condenses into a compact object, which then lies along the inside of the nuclear envelope. As shown in the picture of that red cell, the bar body is that yellow highlighted area of the inactive X chromosome. So this condensing will inactivate or silence most of the genes on that specific X chromosome. Uh, this happens when the zygote is only about 32 cells large, and one of the two X chromosomes is then chosen at random to then be replicated and passed down to subsequent generations. Okay, so this process is seen in calico cats. In cats, fur color is located on the X chromosome. X chromosome inactivation occurs during embryonic development, which then results into tortoiseshell pattern if the cat has two different alleles for this coat color. Since this is an X-linked sex trait, male cats do not express the tortoiseshell fur coat seen in calico cats. However, the process of X chromosome inactivation will result in tortoiseshell fur color in female cat offsprings, in female cats that are offsprings of a cat that is heterozygous for this fur coat trait. So due to this process, some female cats will have the tortoiseshell fur color and are known as calico cats. So essentially, the allele for black or fur is condensed on an X chromosome and becomes inactivated, allowing the other allele on the other X chromosome to be expressed. However, these cells will continue to be chosen at random during embryonic development, allowing for the expression of orange and black fur patches. So, this is depicted in this image on the right, where let's say like half the cells will express orange fur coat color because the black fur that will be condensed on one of the X chromosomes, and then the opposite will happen on the other cell. So the orange fur coat gene will turn into a bar body, allowing the black chromosome fur to be expressed. As you can see in this cute cat, where it has orange and black fur patches. So in conclusion, here are a few main points from my presentation covering Module 4. So first, the cell cycle is a series of events that leads to cell division and is characterized by four distinct phases. Next, we discuss the G1 checkpoint, which separates the G1 phase from the S phase and allows the cell to determine if it is ready to start duplicating its chromosomes, as well as ensuring that the cell's internal components are healthy to divide. Next, remember that mitosis is an important phase in the cell cycle that involves the process of cellular division, where a cell splits into two daughter cells. Mitosis is essential to cellular reproduction and allows organisms to be comprised of multiple cells, thus being multicellular organisms. And then we discussed binary fission, in which bacteria and other prokaryotic cells divide into two identical daughter cells and are able to rapidly divide um, as compared to eukaryotic cells. Also, remember that genetic traits can be inherited via sex chromosomes, resulting in offspring that carry traits from the mother or the father, depending on which traits are present on which sex chromosomes. Basically, some traits are found on the X chromosome and some are found on the Y chromosome. And then we concluded talking about X chromosome inactivation as an epigenetic process that causes the inactivation of one of the two X chromosomes found in female mammals, resulting in the formation of Barr bodies and the silencing of some X-linked chromosomes. A good way to remember this concept is to think about all these calico cats and how their tortoise shell fur is only expressed by female cats who have experienced X chromosome inactivation. And lastly, let's answer a few questions. So first, what stage follows anaphase during mitosis? Second, what are the differences between animal cellular mitosis and plant cellular mitosis? And third, what happens when a cell fails the G1 checkpoint? Okay, so feel free to answer these on your own and post the responses in the discussion board post. And here is my reference list, including the citations, for all the information and images shown in this presentation. And of course, thank you for listening to my presentation. I can't wait to watch all of your guys' presentation, and I hope you have a great day.